Okay, perfect.
Forward, backwards, and
Okay.
maybe hit that and it goes off. But
Yeah.
Okay, so welcome to this morning's Advanced Plant Growth Seminar. First of all, I should say that this meeting is being recorded. So if you don't want your image recorded, please ensure your camera is switched off. Our speaker today is Marcus Tiger from the University of Vienna. He did his BSc in chemistry at Hanover before doing his PhD at Gasleben, working on the regulation of photosynthesis. He then moved as a Humboldt scholar over to Dallas in Texas, where he continued working on bioenergetics, working with mitochondria, before returning to Vienna on a Marie Curie scholarship on plant stress signaling. In 2000 on yeast stress signaling. In 2002, switched over to plant stress signaling, with a particular focus on the role of protein phosphorylation and other PTMs. And since 2004, he's had an independent position at the University of Vienna. And today he's going to talk to us about calcium and protein kinase signaling. Donna, could you just confirm that you can see Marcus's slides?
No, we can't. Not yet.
Okay.
The force does not work.
Can we see the slides now?
Yeah, we can see them now. It's looking good.
Okay, so I'll hand over to Marcus. Okay.
Or did you do
I've started the slideshow.
Okay, good. Okay. Where do I do?
That's the question.
Almost. Okay. Good. Okay. Good morning, everyone. Thank you for inviting me here and giving me the opportunity to show our data. I've a little bit combined some things, some topics I thought that might be interesting for you. So I will introduce at the end of my talk a little bit the ADAPT project, where also the James Hutton Institute is involved in with Mark and then Rob. And the long-standing research interest in my group is stress acclimation of plants, particularly protein kinase and calcium signaling. And here I will present data that we generated in 2 other EU projects, in 2 ITNs, in the ITN Merit and Calypso. So, oh, okay. So then I do it from here. No. This is not working.
Let's try to start the slideshow again. Oh, no.
Okay. So well, basically, when plants need to respond to environmental changes, these can be very fast ones, such as intensities in light. If a leaf is then suddenly exposed to shade, or some other stress situations, such as abiotic stresses, heat, cold stress, or drought or pathogen stress, we are interested in how this signal is being recognized by the plant, and then which signaling pathway is initiated to finally mediate the acclimation response. And for this, we need to transduce the signal mostly to, for example, to the nucleus to initiate gene expression, or to some protein kinases to affect to regulate their enzyme activity of downstream components. And of course, these things have a severe impact with the problems of climate change, which are estimated to sum up to 65 billion per year by the end of this century. And this is what we are doing in the ADAPT project. And this is not only important for us as scientists or for economy, but also the kind of end users of our knowledge, the farmers, feel already this pressure. This is some data that we generated in the ADAPT project that were done by the AGES, the Austrian agency for food and health security. And they carried out a survey for more than 500 farmers all across Europe. And it was interesting to see that they said over the past 10 years and also in the future, they think also drought and heat stress will be the most threatening issues for the potato production. So coming back to this signaling, so I will first now talk a little bit about the molecular mechanisms of signaling of acclimation to stress. And this involves calcium signals and protein kinases to initiate a cellular response mediating the acclimation in the end. And here we are talking about different very well conserved protein complexes. So because very often the question is, the first question the cell needs to answer is to grow or not to grow. And this is mediated by these two ancient protein kinase complexes. So as a promoter of growth, we have the TOR complex, the target of rapamycin, which is promoting cell division, protein synthesis, and so on. So under optimal growth conditions. But under stress conditions, this SNRK or AMP kinase complex becomes activated. And this is very highly evolutionary conserved. So in mammals, we have this AMP activated protein kinase complex. In yeast, we have the SNF1 for sucrose non-fermenting. This indicates this has already something to do with energy signaling. And this is also conserved in plants as a SNF related kinase complex. And we can trace it back to Chlamydomonas and some things even to archaea, some regulatory compartments. So this indicates already, as it's written here, that this core stress signaling pathways involve protein kinases related to SNF1 or AMPK. And this suggests that stress signaling in plants evolved from energy signaling in an evolutionary view. And if we now look what is downstream of this SNRK1 complex, we see there are metabolic enzymes that are directly regulated in their activity by protein phosphorylation. We have also transcriptional factors, transcriptional regulation, because we know that the downstream of the SNRK complex in data from Elena Baena Gonzalez, it was shown that more than 1,000 genes respond to this complex. And very early on, there were some indications that these BZIP transcription factors are downstream. These data came from protoplast experiments, as shown here, and also from the diurnal analysis of gene expression, where also then these BZIP transcription factors became interesting candidates, downstream candidates of the SNRK kinase. So BZIP stands for basic region and leucine zipper. So these BZIP transcription factors bind DNA as a dimer. They bind with their basic region to the DNA, and they have these zipper domains, this leucine zipper domain for dimerization. And also these are highly conserved. So already in yeast we have 14. In higher organisms we have even more. In Arabidopsis we have 75, and in plants so we have usually between 60 to 70 or some more of these BZIP factors. And they have very prominent roles. So we see they are involved in developmental regulation, for example, with high 5. We see they are also involved in ABA, stress signaling, with this AREPS or ABF. So these are also members of the BZIP family, or also in a floral development with FD, which is also a BZIP transcription factor. And we got interested in this subgroup here, in this subgroup C, which forms heterodimers with the subgroup S, because we fished this BZIP 63 as a potential target of the calcium-dependent protein kinase. And we wanted to know if this is also true in planta and what the meaning of this is. So therefore, the first question was, is this also, if it's a target of the SNRK1, is it also involved in energy signaling in metabolic regulation? So we obtained knockout mutants of this BZIP63, and we saw that this under starvation, so in dark-induced senescence after 9 days, has a kind of stay-green phenotype, whereas the overexpressor lines had a faster senescence. And this phenotype could be rescued by addition of sugar. So if we germinate them if we grow them on plates and adding glucose, then this senescent phenotype, for example, this is rescued again. So this indicates that indeed the energy status of the cells is transmitted or transduced to signal by this BZIP63 transcription factor. The next question was then, is this really dependent on the phosphorylation? Because if it's a target of a kinase, then it should be phosphorylated. So we made use of a GFP fusion of this BZIP63 that we were able to detect it, because one problem is that they are really very low abundance, these transcription factors, so they are very hard to detect with biochemical assays. And we looked, we made a 2D gel and saw that we get a certain spot pattern after Western blot of the 2D gel. And if we treat the sample with lambda phosphatase, we can shift it. So that removes the phosphorylation, and that shows that there is indeed some phosphorylation going on. And the question was, is this now dependent on the energy status? And for this, we use this extended night treatments. So what you see here is the normal day-night cycle as an accumulation of starch during the day, which is then used up during the night. And at the end of the night, there is no starch left. And if you then from one day to the other extend the night for several hours, the plant is not prepared for this. So the starch is left, so it runs into energy problems here if it stays now a few hours more in the dark. And we did this, and we applied here because these 2D gels are very difficult to run, and if you have several samples, then it's much easier if you can have another one-dimensional separation, and therefore we applied this Fostech technique. So you have this Fostech reagent, and this Fostech reagent binds to phosphate groups, so to phosphorylated proteins, and this leads then to a better separation of different phosphorylated forms of proteins. So if you have a protein 1 or 2 times or 3 times phosphorylated, it will migrate much slower in the gel. And when we did this, here, for example, is a recombinant BZIP, which is not phosphorylated. We saw that we see indeed different patterns here. So we have differently phosphorylated forms. This can be removed by lambda phosphatase treatment. So it's indeed a phosphorylation where we are looking at. And we see there is clearly a difference between light and extended night treatments. So where we see a form coming up here and all over the, if we look at the entire pattern here, we can separate 7 different bands here in this assay. And this is clearly dependent on the energy and on the night or day length conditions. So we said, okay, what are these forms and where is this protein phosphorylated? So we wanted to know where are the phosphorylation sites. So again, we made use of this BZIP63 with this GFP fusion, because with this GFP fusion, you can use this trapping beads, so which are working very well for immunoprecipitation. And you can see here with the antibody, but even in the silver stain, you can see you can precipitate really a significant amount of BZIP protein. And that's absolutely essential if you want to do proteomics and phosphoproteomics, you need some level of proteins that you can really see. Yeah, so this was very important. So we could do this IP, and we could identify 1, 2, 3, 4, 5, 6, 7 phosphorylation sites. So that's exactly matching these 7 bands what we have seen before. Yeah, so this tells us indeed under in vivo condition we have 7 different phosphorylation sites distributed over the protein. What was very important here is normally if you do proteomics, you do just a trypsin digest. And then you see here, and then you get this peptide, you get this, you can cover this area here, this area, this and this area here. And you see, okay, we were very lucky because this serine 29 is very important. We will see later on we had covered this. We had also covered here this C-terminal part, but for example, we did not cover this serine 160. I will come to that later. And what you also see is that here, for example, it's not found to be phosphorylated in many samples, and some it is. Yeah? The same is true here, whereas these here are mostly, when we identified those, they were found phosphorylated. Yeah? The same was true here, but there was only one digest that allowed us to detect this peptide. So the next question was then, which kinases do phosphorylate these BZIP factor? Because with 7 different sites at different places, so this indicates that maybe a lot of different kinases do this. And actually, this was done many years after I pulled out the BZIP63 and AGES2 hybrid as a target of CDPK. But then we found in the CDPK mutants, there was not really an effect that we saw. So we said, okay, maybe we should have another unbiased look at this. And so we did this in-gel kinase assays. So for this, you have a normal SDS page, and you co-polymerize your substrate into this gel. So for this, you need really milligram amounts of the substrate, in this case of the BZIP transcription factor. It's co-polymerized in the gel, so it's everywhere. Then you load your total protein extract to a normal SDS page. Then you wash out the SDS. The protein stays in, and then you add hot ATP, and at the molecular weight of the kinase, then you get a phosphorylation band if it's phosphorylating BZIP. Yeah? And when we did that, it looked like this. So we saw 3 different bands at the minimum. So we saw, okay, there might be 3 different kinases that phosphorylate this BZIP protein. Now this explains already a little bit why we saw these many phosphorylation sites. So the question was then, okay, what are these kinases? So we did an affinity purification. So we immobilized the BZIP63 as a GST fusion on a GST trap column. Then we put plant protein extract on this, washed it with increasing salt concentration, and then we could massively reduce the complexity of the sample. And then we did this in 2 gels. So one we did the kinase assay, and the other was without substrate, of course, where we loaded the same sample, and then we excised these bands there where we show where we could see the phosphorylation of the target, and identified these by MS. And we can see that indeed we can recover these protein kinases in samples with a low complexity, and there we identified now the SNRK1. So in fact, this was our way how we identified, okay, the BZIP is the target of the SNRK1, but we found also the CDPKs. We also found the CPK3 here. Yeah, so we see already here that we have different targets, the different kinases targeting the same transcription factor. And we saw also a casein kinase here. Okay, so we wanted to see, is it true or not? Is this a bona fide target or not? So what about this SNRK1 complex? So do we see colocalization? That's the first thing. So the BZIP transcription factor is always in the nucleus. So here you see infiltrated tobacco epidermal cells. So you have a nuclear signal for the BZIP. And you see also the AKIN10 is there. That's a kinase subunit of the SNRK1. There's another one that the akin 11 is also there, and the regulatory subunit SNF4 is also there. So they co-localize in the nucleus. That's good. They do also interact in the nucleus if we do a split YFP or split CFP, where we fuse the BZIP63 to one part of the CFP and the other partners to the other part, to the C-terminal part. And then we see co-localization and interaction in the nucleus. Here, this is a dimerization, homodimerization of BZIP63 as the positive control. We see a clear interaction with kin10, almost nothing with kin11. There's also interaction with SNF4. And we see the same thing in these two hybrid assays. Also here, so this would be the positive homodimerization of BZIP63. Here would be the interaction with kin10. Here there is nothing detectable with kin11. And here this would be the regulatory subunit SNF4. So there is co-localization, there is interaction. So the question is, what if we knock out the kinase? So we obtained a BZIP63 knockout line, and we can see that we can see this phosphorylation pattern here in vitro assays. Kin10 does nicely phosphorylate it, kin11 very weakly in vitro. And if we now do these extended night treatments and look for the band shift in the Fostech gels, we see here is the extended night of the wild type, and here in the akin10 mutants, it's almost gone. So then reviewers said, well, but there's still a little bit of shade here, as a akin10-11 double mutant is lethal, so you have to use some line, for example, by virus-induced gene silencing to knock out both of them, but then you can also completely block this shift. So this tells us now, okay, so indeed, kin10 is really one of the kinases that's regulating this BZIP in vitro, but which of these sites here are affected by this? So this was then the next thing, what we did, that we did point mutations, and we used the in vitro kinase assay, where we have the recombinant BZIP, and putting this together in an Eppendorf tube with the kinase, and mutating now all these sites. And you see here, this is the wild type situation. Here you see with the serine 29 that the phosphorylation is already weaker, and here in this lower part they are completely gone. What we always saw, and what you see here on the COMESI, that we have these 3 different bands, and this is just due to the effect that we have always C-terminal truncation that happens if the protein purified. It's purified via the GST tag at the N-terminus. So this is here the C-terminal degradation that happens during this purification process. And you see we have, for example, these 2 phosphorylation sites in the C-terminus. Yeah? If they are not here in this degradation product, then also you do not see anything here. Yeah? And you see when you have other mutations, there is no effect. So many of these sites are not targeted by SNRK. And if you mutate the serine 29, 294, and 300 to an alanine, then the phosphorylation signal is gone. So these are the 3 sites. So they are phosphorylated. The question is now, what does it mean? Because this was a little bit, it was nice to see it so clearly, but looking for the function, it was a bit disappointing, because if you look at the BZIP factor, we have the DNA binding domain here, we have the zipper domain here, and we found the phosphorylation site at the very end in C-terminus. So far away from the really interesting region, so to say. Huh? So does it mean something or not? And therefore, we had a look at all the BZIP sequences from Triskomitrella rise to Arabidopsis. And of course, this is highly conserved. Here, this serine 160 is always a serine in all the species, but also the other sites that we found, the serine 29 is always a phosphorylatable residue. It's always a serine here, and the same is true for the C-terminal part. Yeah. So we thought, okay, if it's so highly conserved during evolution, then there must be a function for this. So we took this BZIP63 knockout mutant that I showed before, and did a complementation using the endogenous promoter and genomic BZIP protein fused to YFP, and we mutated this serine and these serines here to an alanine that cannot be phosphorylated. And then we look again for the phenotype. So we see there is this stay green phenotype of the knockout in dark induced senescence, and this is rescued in this mutant, but this is not rescued. So if we mutate, if we complement with the wild type allele, but if we have this non-phosphorylatable form, it is not rescued. Yeah, and the same is true for gene expression. So I did not show all these data. We saw in different metabolites, we see a very strong accumulation of sugars in the BZIP63 mutant. This is reversed in the wild type complementation, but not in the non-phosphorylatable form. And here is the overexpressor where it goes in the other direction. And we can also pin this down to certain marker genes that are expressed. So, for example, the asparagine synthase, it's also called DIN6 for dark-induced number 6, which is strongly induced 4 hours after exposing plants to extended night in the wild type, but not in the mutant. And if we then bring in the wild type allele, it is complemented, but not by the non-phosphorylatable form. So we need this phosphorylation. The question is for what? Because it cannot be the DNA binding. It will most likely also not be the nuclear localization sequence, because that is also in this BD domain. So we thought it's maybe the dimerization. And therefore, we did some yeast to hybrid interaction, and we did also protoplast to hybrid interaction, and it indicated that this is the case. And then we thought, I thought this is a very crazy experiment, but maybe let's try if it works. So we just thought, okay, maybe the phosphorylation will affect the choice, the preference of the dimer formation in the plant. So we said, okay, we take the C-terminal part of BZIP63, fuse this to CFP, and then we offer the N-terminal part from BZIP1 or from BZIP63. So that we can see, okay, under certain conditions, do we rather see the homodimers or the heterodimers with BZIP1 or the heterodimers with BZIP11? So homodimer formation we cannot see here in this assay. And then we co-express the akin10 in a protoplast. And what we see is there is a clear shift in the dimer formation if we have the non-phosphorylatable form compared to the wild type form. So this does clearly show that also in vivo implanter, the phosphorylation by the kin10 does change the dimer formation of the BZIP factors. And this has a consequence. So the model is that then under stress conditions, we have the phosphorylation. We know in the meantime that it's mostly the phosphorylation of this serine 29. This is, by the way, also an interesting take-home message. There are 7 phosphorylation sites in that protein, but under this condition, for the effect we see here, only 1 site is really important. Yeah? And in this case, that's the serine 29 that we know in the meantime that is then changing the dimer formation from the homodimer to the heterodimer and the other way around. And we know from other data where we co-expressed different combinations of BZIP factors in protoplasts, and we looked at the gene expression patterns, we see that completely different sets of genes are induced if we, for example, compare the BZIP1 plus 63 with the BZIP B63 plus BZIP11. So we have some genes are, of course, similarly induced, but there is clearly a difference of certain genes that the dimer formation makes a difference for the downstream gene expression. And I showed you before that we have not only one kinase, so we have at least three. So I showed you now the data for the SNRK1. I told you already that I had fished the BZIP63 as an interaction partner for a CDPK. And we see here that we have a different phenotype. Now we have salt stress sensitivity, which is rescued by overexpression of these BZIP factors. And this fits to the published phenotypes that we published before for the CPK3, that also the CPK3 mutants, here is a knockout mutant and here is an overexpressor lines, are also salt sensitive. So also here we made the same experiments. We looked for the co-localization with the CPK3. It's a bit more complex, but we can clearly see it in the nucleus. Here in red, you have the chlorophyll autofluorescence from the chloroplast, again in epidermal cells. And we see also yeast to hybrid interaction, quite strong, similarly to the one that we see for the homodimerization. And we see also in the BIFT assay, the bimolecular fluorescence complementation interaction in vivo in the nucleus. So now we made the same experiment again, what I showed before, where we wanted to see which of these amino acids is now phosphorylated. So again, here are the bands you see from the wild type. This is degradation products. Now you see here there is no difference in all these sites. Only here is a clear difference in 160. And there is another one if you combine this with the C-terminal sites. So from this we see there are different sites that are phosphorylated, and what was really important for us is that these 160 is targeted by CPK3 because that is a site now that is clearly in the key DNA binding domain of this BZIP factor. And I showed you already before that this is highly conserved, of course. And what happens is if you phosphorylate this site here in the DNA binding domain, then it will just disturb the interaction here and DNA binding. So therefore, the DNA binding is strongly lost if you phosphorylate it or if you mutate these residues here to a phosphomimicking version. So altogether, we see a very complex picture here, where we see that we have a number of phosphorylation sites at different transcription factors, and these are the reactions that occur in the nucleus. So where the transcription factors are phosphorylated and then, for example, change their dimerization patterns. We have also seen in other phosphoproteomics analysis. Here in this paper that a set of enzymes, metabolic enzymes in the cytosol is phosphorylated. And what was also very interesting for us is that we could in the beginning not understand why we see differences also occurring in the chloroplast, for example. That at some point also phosphorylation events in the chloroplast were affected. Although we are quite sure that the kinase is not there, but there are a number of chloroplast and mitochondria targeted genes that are direct targets of these BZIP transcription factors. And now what we want to do a little bit, so the outlook for the ADAPT project, I mentioned already the FD, which is also a BZIP transcription factor, and is indicated here. Also here we need to have dimerization. So far, it's not really clear if it's always homodimers or heterodimers and if different dimers do different things. But what it is known, at least very well known from Arabidopsis, that the FD is phosphorylated and then 1433 protein binds to these phosphorylated forms and initiates this complex for transcriptional activation with FT. And this seems highly conserved. So this is also in potato the case for flowering and also for tuberization. And also here, different CDPKs play an important role, but it's not that simple. It's not only one. The CPK, what I just showed, the CPK3 has a phenotype that's related to flower initiation, to flowering. So this is a bit misleading because this CPK3-2 is a tDNA insertion line, but it's an insertion in the promoter which leads to a little bit higher protein expression. So this is a slightly overexpressor line, and here is a mutant, and this is a higher order mutant because here we have now the problem that we have 34 different CDPKs, and you have really to knock out a lot of them to get to a very strong phenotype. So, but we have seen this is the case here for this quadruple knockout that we received from Julian Schröder for this CDPKs. So the question is now, what are the functional organogs of these in potato? So we have cloned these and we see that some of these things are affected. And we think that they play similar roles in tuberization and maybe also in stress. And this leads me now to this ADAPT project, where we have the long-term aim to accelerate development of stress-tolerant potato. And this project covers also the whole range from field work to lab work and asks specific questions. We have a complex network, so we have a huge group of research groups from different universities or research institutes like this one. And we have teamed up with potato breeders. So we have also very active potato breeders for this stress trials with HPC and Maya. Solana is mostly doing transformation for us because we want to use these new lines that we want to establish for our research. And we have also a strong link to the regulatory agencies and also to PSI, a technology developer. And I will talk about this link in a moment.